Start off. So I would like to start by thanking Deepa and Kavita before I forget for inviting me here and letting me present my work. So far you heard like really cool stuff about epistasis and all that. Now we'll go back to a classical problem, that's of speciation. So I work on molecular mechanisms of speciation, and in this talk, I would like to present some of my work from my postdoctoral lab, which I performed, wherein I used yeast in order to identify how speciation happens, in general, how postzygotic isolation really comes in, what are the factors which come in. So since I said it's yeast, it's a eukaryotic species, and the basic definition of a species in eukaryotes is that there are groups of interbreeding natural populations that are reproductively isolated from other such populations. In other words, it's this reproductive isolation which prevents the gene flow between two populations. And there are two forms. If I'm going too fast, please ask me to slow down. And if you have any questions, you can always stop me at any point of time. That's not a problem. So there are two forms of reproductive isolation. One is prezygotic reproductive isolation, which basically means zygote is not formed. Before the zygote itself is formed, it's isolated. Second is postzygotic, which means after zygote formation. So we look at some example. For example, the prezygotic isolation, it could be because of mating discrimination. For example, in the case of the left and right-handed snail, unsuccessful gamete formation is also another popularly known cause of prezygotic isolation. In the case of postzygotic isolation, it's characterized by its hybrid viability or sterility, inviability or sterility. The famous example is that of horse and donkey, which gives a mule. Mule being robust, it just does not reproduce, and so it happens. So yeast, which is my favorite organism, these are called sensu stricto yeasts, and I've not put all these species. It's an old model. There are several other species which have been identified in between. So it's an old model, but this is good enough for our talk today and our work right now. So these yeasts are supposedly under postzygotic reproductive isolation. And the yeast which I am going to show in this talk is Sacchromyces cerevisiae, which everybody probably knows. And its neighbor, which is diverged by around 20 million years, it's called Sacchromyces bayanus. So when I say it's under strong postzygotic isolation, the immediate question would be that if it is like mule, how on earth can you continue working with evolution or do experimental evolution or any kind of sexual evolution with this kind of organism? The reason is that these yeasts, though they are hybrids, they can reproduce asexually without any obvious defects. And in case of meiosis, that sexual offspring, you have 0.5% viability. If you take the sample size being very, very large, 0.5% is good enough to have your viable offsprings in order to continue working with it. And these two yeasts are also sympatric. So it basically means that they have the same number of chromosomes, very, very similar characteristics, except for its growth temperature and all that. For example, bayanus likes to grow at 18 degrees, and cerevisiae, on the other hand, likes 30 degrees. So there are some differences, but basically they both are budding yeasts, and you can work with it more comfortably. Okay, so there are like three major mechanisms of postzygotic isolation, among which genetic incompatibility is known to be a very important driver. It's considered as one of the main drivers of postzygotic reproductive isolation. Okay. So what is this genetic incompatibility? This was proposed by the model, Dobzhansky-Muller model of genetic incompatibility, derived from yeast crosses, which around 1940s, they came up with this model. Now let us look at this carefully. Now suppose you have an ancestral genotype, in which you have two very important genes which are interacting with each other. They are very important, keep that in mind. So you have capital A and capital B, which are two very important genes which interact with each other. After some time, due to some reason, they separate out as two subpopulations. And there's a mutation which changes big B to small b. And here, in the second population, A is changed to small a. Now, since they are important genes, they just cannot get rid of it, and they start co-evolving with each other. And now B is comfortable with big A, and big B is comfortable with small a. Okay. So after some time, if these two come back together, now this interaction between small b and small a is not is not going to be viable anymore, and that's basically what is called as a genetic incompatibility. In other words, there are alleles that are normal in native populations, but are deleterious in diverse populations. So this is the idea of genetic incompatibility. So there are several studies which have tried to look at genetic incompatibilities. Some of them are also from my former lab, and we found a few mitochondrial-nuclear incompatibility. So again, taking the same two species, cerevisiae and bayanus, if you take the AEP2, which is required for ATPase expression, which is encoded on chromosome 13, so this is a nuclear encoding gene, and OLI1, which is required for oligomycin resistance, is a mitochondrial gene. Now, in these, bayanus and cerevisiae, they have their own, they have co-evolved with each other and they work well with each other. So when you have a hybrid, this bayanus, AEP2, cannot work with cerevisiae anymore. So it literally kills the cell. It's a very strong mitochondrial nuclear incompatibility. It's just as one example. Several other organisms have been used, and they have found strong incompatibilities in yeast, C. elegans, fly, Arabidopsis plant, mammalian systems such as mouse model, etc. So several of them have been found. And all these incompatibilities which have been really, the molecular mechanisms which have been studied are two loci incompatibilities. Okay. But what about weak incompatibilities? Weak incompatibilities are a little bit different from the strong incompatibilities, right? So here you have a strong phenotype, you can identify it, you can dig into the molecular mechanisms. But do we have to study weak incompatibilities in the first place? So let us first see what weak incompatibilities are. Now suppose you have this, these are your chromosomes and you have some genes which are from a foreign species. Okay, this is a hybrid. So you basically have a hybrid system here. You have Y, X, Z, and M. Y, Z, and M are from one species, whereas X is from another species. Now individually, they don't show a very strong phenotype. Okay. On the other hand, when all three of them occur together, they can be deleterious, strongly deleterious. They might have a very strong phenotype. That's basically how we define a weak incompatibility. Okay. Now, why should we study these weak incompatibilities? There are several models. The model which was presented by Presgraves and et al. Is something like, it's very important for the incipient stage of speciation. So now, you have a weak incompatibility, and over a period of time, this can become stronger. I can't see how long I'm going. Yeah, that's right. So, over a period of time, these weak incompatibilities can become strong incompatibilities and thus cause speciation. That's one possibility. Second is that you have a weak incompatibility, so it accumulates a compensatory mutation which somehow benefits, and later on these mutations become a different phenotype, or it gives you a different function which is not there in the other population. So when they come together, they have a problem. They can't work with each other. Either ways, if you understand how these weak incompatibilities work, you could probably understand how speciation happens, or like how speciation really arises or like starts off in the first place. And when you look at the experimental crosses across, like all the experimental crosses which have been done, several cases have been observed, but we do not know what is the consequence or how, or what is the molecular mechanism of the system. Okay, so we wanted to actually ask this question. How do weak incompatibilities cause speciation? That's the first question which we wanted to ask. And what is its consequence in hybrids? Okay, so these are the two main questions which we wanted to ask. So I showed you a mitochondrial-nuclear incompatibility in yeast. So for some reason, nuclear incompatibilities have been elusive in yeast. We have none of us, there's only one case of SIR2. We don't know the molecular mechanism yet. We just know that, okay, there is a possibility that there's a nuclear incompatibility. Otherwise, we don't see nuclear incompatibility. So we also wanted to see that if you have weak incompatibilities, probably in the nuclear genome, you have a higher chance of these weak incompatibilities, and we have not been able to identify till now. And if we can figure out what is happening here, we could also resolve this problem. So we kind of clubbed this also into this set and wanted to see what is going to happen. Okay, am I clear till now? Am I going too fast? If I'm going fast, please ask me to slow down. Yeah, okay. So for this, we use something called the chromosome replacement line. I think you should focus a little on this slide because I'll be using these numbers and replacement lines over and over again. Okay, so these chromosome replacement lines are basically introgressed hybrids. So what we have done is that there are 16 chromosomes in Sacchromyces cerevisiae or in Sacchromyces bayanus. So we replace one chromosome of Saccharomyces cerevisiae by its bayanus counterpart. So you'll have a 15:1 combination most of the time. Sometimes you do have a 14:2 combinations. Now, if you have like 1 here, it means chromosome 1 has been replaced. The number here designates the chromosome which has been replaced. 3 means third chromosome has been replaced. Okay. Now, sometimes you see 5 plus 7, 6 plus 10, and 8 plus 15. We know for sure that 6 plus 10 and 8 plus 15, it's because of translocation. There's a translocation in bayanus. So you need to have, there are essential genes with this translocation. And if you don't put these two chromosomes together, because they are essential genes, the cell dies. So we have to keep them together. In the case of 5 plus 7, it's still unclear exactly why, but we still don't have viable cells if we don't put 5 and 7 together. It's surprising that 7 alone can stand, but 5 requires 7 to be there. It's still not known. That's a problem which is still open. Anyway, so what is the advantage of using this when compared to complete hybrids? Now, what is the advantage of using this system? Because they are introgress hybrids, right? And most of the studies, they use complete hybrids. Yeah. So when you're using complete hybrids, one of the biggest hurdle you have is the statistical power and the epistatic load, which is corresponding to that. So you have a lot of interactions, a lot of noise, and finding the signal from this noise is always difficult. So speciation studies find it very difficult to figure out what is happening. On the other hand, if you're trying to look at the other molecular mechanisms of postzygotic isolation, like sequence divergence, maybe complete hybrids are the best things to do. But if you're looking at genetic incompatibilities, finding certain loci, it just doesn't work. Okay, so you have each of these introgress lines can give you a chromosomal level understanding of what these incompatibilities or genes are doing. So that's one big advantage. So you have a higher resolution or picture of what these genetic incompatibilities do. Second is that when you change one chromosome, you are basically having a new genotype. So you have several kinds of genotypes, and if you're trying to do an evolution experiment, this is like you have like 16 genotypes or 11 genotypes, and you can evolve it and see how different genotypes are going to change. And there are not major differences between them. There's one chromosome which is big enough, but it's not like two different species altogether. So you can start seeing how introgress hybrids really evolve and all that. So that's one advantage. Another advantage of using replacement lines. So we use these replacement lines, and the first question which we wanted to ask was that, okay, what is the consequence of a foreign chromosome on its parental genome, like Sacchromyces cerevisiae genome? So I have 15 chromosomes here, one chromosome coming here. How does it really affect? To do that, we used RNA sequencing, which is one of the more popular approaches, looked at the transcriptome, and we used log phase cells and cultured at 23 degrees. The reason why we chose this weird temperature is because it's in between 28 or 30 degrees and 18 degrees. So we are not, even though we are only putting one chromosome, we don't want to say that, okay, there's a bias of temperature when you're culturing them. So it grows well, no problem. Okay, so what we observed was that there's a set of data called environmental stress response data, which was published by Agashe. So I call this as a yeast abuse experiment. She literally abused yeast with very severe conditions. Okay, and she found that when you abuse them in different conditions, there's a common set of something like 900 genes which get commonly misregulated in all these conditions. And that's your environmental stress response data. So whenever people are trying to look for stress signatures, one of the things they do is that they try to see if their transcriptome is somehow correlated with this set or not. If it is correlated, you have an idea that, okay, they have a very similar trend of environmental stress response, so there must be a stress response. And when we took our cells, which were cultured at very normal temperature, the only anomaly there is a foreign chromosome, we find a strong correlation. So this correlation confirmed the idea that these cells are under stress. It's not surprising. Okay, fine, you have a hybrid, it's under stress. So what kind of stress is it? Stress is like a very big term. It can be like anything, right? So we wanted to go and see what is happening. So when we were thinking about this, we came across this study by Angelica Ammon. So she used something called as a disomic cell. It's actually an aneuploid, but what she did was that it's a haploid cell in which chromosome has 2 copies, one chromosome. Say chromosome 1 has 2 copies, or chromosome 2 has 2 copies. So it's disome because there are 2 copies of a particular chromosome inside the cell, while all the other copies are euploid, no problem there. So it's an aneuploid cell with 1 extra chromosome. Okay, so she used this and she found that these aneuploid cells suffer from proteotoxic stress. Now, what does this mean? It means that the stoichiometry of protein homeostasis is disrupted. So in a wild type cell, you have the right number of copies for it to interact. So everything is fine. But in the case of a disomic cell, so extra copy, one whole extra copy of this chromosome is being present inside the cell. So they start lying around in the cell and they cause overloading of the proteasome degradation machinery, causing proteotoxic stress. So this was our finding. So we had this idea, okay, so they also found that proteotoxic stress suffering cells also have ESR signature, the environmental stress response signature. So we wanted to see, okay, our case is not very different from theirs. They have extra chromosome, we have like -1 chromosome because it's a foreign chromosome. If we assume that some part of this chromosome genes are not working or proteins are not working, it's more like negative towards like reduction in the number of proteins than more proteins. So you still have many of the partners which are lying around, so it should have a problem. So that's one possibility. The second possibility is loss of interaction completely. So since we are putting only 1 foreign chromosome and keeping the rest of the genome from cerevisiae, if there is loss of interaction in the hybrid, in such a case, you will have the whole set going out, and basically that also can overload the proteasome machinery, proteasome degradation machinery, and you can have proteotoxic stress. So either ways, there's a chance that you have proteotoxic stress. So we wanted to test whether this is meaningful or not. So in order to do this, what we did was that we used a very classical assay with HSP104. This is a marker, this is a disaggregase, it's a protein disaggregase. So when you have misfolded proteins, they usually form aggregates inside the cell. They are not just allowed to like float around because it kind of impedes the cellular machinery. So they're held as aggregates, and when they're supposed to be degraded, this HSP104 goes and binds to it, comes to its active or functional form, and opens these aggregated proteins and sends it out for degradation. Okay, so now we tagged it with a fluorescent protein, and we wanted to see whenever you have an aggregate, if when it goes and binds to it, we can see a dot or a foci. So if you see a foci, it means you have protein aggregates. More number of protein aggregates basically means that it has more cells, which are probably more proteins which are non-functional in the cell, so with higher chance of proteotoxic stress. Okay, so now this is the wild type Sacchromyces cerevisiae. And we started it to 23 degrees. You don't see any foci here. And then we moved it to 37 degrees and we did a time series experiment. Every 45 minutes, we look at, we take an image and then count how many foci they have, or like how many cells have foci. That's what we are more interested in. Okay, now if you look at the other replacement lines, for example, replacement line 16, which is chromosome 16, which is being replaced, or 8+15, both these chromosomes that are replaced, even at 180 minutes, you can see that the cells are still harboring a lot of protein aggregates. Okay, this kind of quantified the fact that you have, these cells are probably suffering from proteotoxic stress, and the protein degradation might be hampered. Okay, so we quantified this data, and this is the total data. So now, these are the percentage of, the y-axis here is the percentage of foci containing cells at 180 minutes. That's the last point which I showed here, because beyond this, the wild type does not have any foci, we don't care about it. So we actually did it for like 300, 400 minutes, and we saw that slowly they started degrading, but yeah. So if you look at it, we find that this is a cerevisiae. It is having very few cells, like 2% of cells have a foci. When you look at the other cells, all of them have significantly a higher fraction of cells containing protein aggregates. Among them, this 5+7, 8+15, and 16 are drastically higher. These 3 lines somehow harbor more protein aggregates when compared to the others. So that's why I marked it in these boxes, and I'll come back to these lines over and over again. Okay, so 5+7, 8+15, and 16 are sick. So if you have proteotoxic stress, what is its consequence? So we wanted to see by looking at its growth rate, that is mitosis, and sporulation rate, which is meiosis. So when we looked at this, when we culture our cells in YPD, which is the rich glucose medium, without any treatment, we don't see any difference. I've underlined the controls here, that is cerevisiae and bayanus. So we are comparing all this with respect to this right now. We don't see any significant difference. Okay. On the other hand, when I start inducing a mild stress, it changes a lot. For example, I use HSP90 inhibitor. HSP90 is another chaperone which is required for the late stage folding of misfolded proteins. Okay. So when I did this, I start seeing that all the replacement lines are having a significant fitness defect. Okay. So when you look at this, again, 5 plus 7, 8 plus 15, and 16 suffer from strong, very significant fitness defect. So these were the ones which had harbored extreme aggregates too. Okay. So after this, we wanted to, so because you don't see this kind of a fitness defect unless you're inducing it, we thought it should be a weak incompatibility. Otherwise, like if it was a strong incompatibility, you culture it, you see a fitness defect, then it's all because of the presence of foreign chromosome and you don't have to push it. So we found that only when you push it, you're using up all the chaperones, HSP90 chaperones. So now, all those which are misfolded proteins or loss of interaction proteins were now overloading the cell, so you see more protein aggregates. Okay, yeah. So we did another experiment with 32 degrees, which was another mild heat stress, increasing the temperature by 2 degrees than the normal temperature of Sacchromyces cerevisiae, and we found it was highly correlated with that of geldanomycin treatment. So what about sporulation? We found that the same 3 lines on treatment with HSP90 inhibitor were also having sensitivity towards sporulation. The sporulation rate, the longer the bar, more sensitivities, lesser the sporulation rate. So 16, for example, did not sporulate at all. It was just not sporulating. These 2 were the other ones which had higher protein aggregates, and they again did not sporulate well. Okay, so these showed us that if you have probably proteotoxic stress, you have these problems. So can we rescue the fitness of these cells by alleviating proteotoxic stress? So in order to test this, if it is truly because of proteotoxic stress, then if you kind of alleviate the proteotoxic stress, all these defects should disappear, right? So that's basically what we try to do. So we now looked at, there's a gene called UBP6. It's a deubiquitinase, it's a E3 ligase. So what this does is it has dual function. So the way proteasome recognizes the substrates to be degraded is by looking at the polyubiquitin chain. So it looks for this tagged polyubiquitin chain and it then goes for degradation. And this deubiquitinases, they come and cleave it just before it enters the proteasome so that they can be recycled. So you don't have to keep producing ubiquitins. Okay. So UBP6 is not just a deubiquitinase. It has a dual function of, on deletion, it accelerates the activity of the proteasome. Okay. So if I delete this, now proteasome activity will be increased. So all those aggregates which were like lying around should get degraded. So it should kind of alleviate your proteotoxic stress inside the cell. So it should also show that there's improvement in fitness. That's our idea. So we deleted these mutants and we tested for its growth rate. We used these 2 lines which are like extremely defective. I didn't use 5 plus 7 because we later found that the 5 plus 7 also harbors a mitochondrial-nuclear incompatibility. And that somehow comes and interferes with the general fitness of the cell, and so it can cause a problem. That's the reason why I didn't use 5 plus 7 for my rest of my this thing. I focused on 16 and 8 plus 15. So now, if you look at this, the cells were literally dying when I was treating it, and with HSP90, and when I delete it, it recovers a lot. I mean, seriously, it's like many times, right? So the same thing happens with 8 plus 15. So this kind of recovery showed us that, okay, on alleviating proteotoxic stress, fitness defect can also be improved. Yeah. So the next part was the protein aggregation assay, because that's what I started with. I started by saying that you have more protein aggregates, which means that there's more proteotoxic stress. So all these things are happening. So if that is true, the protein aggregate should also be down, and that's exactly what we find. The percentage of cells harboring the protein aggregates reduces drastically when you look at when you delete the UBP6 gene. So, but these 2 evidences are still indirect because it's not directly on the proteasome itself. There could be some other machinery through which it is working. So we wanted to see how would it happen, like if I directly manipulate the proteasome itself, would it make a difference? So we chose a cap component called RPN6, which on deletion kind of, because it's an essential cap component, we did a heterozygous deletion, and the proteasome, 50% of the proteasomes will not work. Only 50% of the proteasomes will be functional. So we are now making the cell, we are giving the cell only 50% of functional proteasome molecules, and we are asking it what is going to happen. So here we expect that the protein degradation should be lower, which means that you should have higher defect. Okay, so when we did this, we didn't use any treatment, no HSP90, no 32 degrees. We just cultured at 23 degrees, and we wanted to see if you delete it, do you have fitness defect or not, and that's exactly what we found. So if you look at this, this is basically without deletion, and this after deletion. You can see that there's a significant increase in fitness defect on deleting RPN6, which means proteasome, if it is not functional properly, protein degradation is affected, which in turn increases the proteotoxic stress, and thus it causes a problem. So all these are more like, we now know it's protein overloading, proteasome overloading and all that. But what is the mechanism? How is this happening? How are these weak incompatibilities coming together to do this kind of proteotoxic stress, cause this proteotoxic stress? When we're thinking about this, we came across this idea that, okay, protein complexes are one scenario where you have multiple interactions happening in one group. So if you have a weak incompatibility, and if there is loss of interaction in a protein complex, then you should have like all the subunits of the protein complex loose in the cell, which is like going and overloading the proteasome. So that must be the cause. So in order to test this, we did a small analysis, and we looked at the number of subunits on each chromosome, the foreign chromosome, and try to see if there are more number of subunits which are forming a protein complex. Is that somehow correlated with the fitness defect altogether? And we find that that's actually true. So if you have the HSP104 foci, you see, or the fitness defect, either case, you find that if you have more number of protein complexes forming genes on a foreign chromosome, they have a higher fitness defect or number of aggregates, which kind of told us that, okay, we could probably go ahead and do this. And we wanted to now extract every protein complex possible inside the yeast cell and look at whether it is stable or unstable in the hybrid. Okay, so we used something called as isotope labeling method, in which we label the wild type cell with a heavy isotope, and the replacement line, which are hybrid, we don't label it. It's just the normal light isotope. We take the cell lysate, mix it one on one, and run it through a size exclusion chromatography. Now, this is a biophysical method in which it kind of separates out based on the molecular weight of the fraction, in the sense that the ones which come out early are heavier, and the ones which come out later are lighter. So the complete complexes can be, you can always expect them to be heavier because there are more subunits, molecular weight will be higher, so they should all come out earlier. And the ones which are not complete, which are unstable, should come out later. Okay, that's the idea. So when we did this, we found that these are like the idea. So if you have a loss of interaction somewhere, they should turn out here, in the right end of the spectrum. Okay, so after this, we will run it through a mass spectrometry, and we can find out what is the relative abundance of each fraction. And we developed an algorithm. I'm not going to go into all those details because of the shortage of time. As to we predicted, or we identified each of these protein complexes corresponding to each of these fractions, identified them, and tried to look at it. Now, what we expected to see was the following. Now, if it is wild type, you would expect most of them to be here because they're all complete complexes. On the other hand, if it is a replacement line, because there is this destabilization of protein complexes, you should see a change in the pattern difference, in the sense that instead of this curve, there should be increase of protein fractions coming off in the lighter fraction or lower molecular weight fraction. Okay, so when we use this pattern difference, now this is the pattern difference, each of them, this is like when you look at the fold change between cerevisiae and the hybrid, This is with respect to cerevisiae and cerevisiae itself. When you look at this, we found that there are 14 protein complexes which turn out to be unstable. There's a large number of protein complexes actually in any cell. And among these, we find many of them are very general, they involve in general cellular machinery like polymerases, 1 and 3, eukaryotic translation elongation factor, transcription elongation factor, that they are required for daily functioning of the cell. And that's the reason why when you increase the temperature even by 2 degrees, which is really slight, the cell dies, it cannot survive it. Okay, so we found that weak incompatibilities can somehow, one scenario is that the protein complexes are breaking down, and these are going and kind of overburdening the proteasome machinery, clogging it completely. So the cell cannot respond, it is sick. So since it is sick, it does not want to use its energy getting into meiosis, because meiosis is very expensive. So instead, it conserves its energy in order to maintain the cellular function. That's how reproductive isolation can form. And weak incompatibilities can cause postzygotic reproductive isolation. Okay, that was the major conclusion which we had. So now, in cerevisiae, this is the model. So we have correctly folded proteins and stable protein complexes. And then, in hybrids, you have loss of interactions and incompatibilities, which breaks these protein complexes, which in turn causes protein homeostatic imbalance. And later on, it causes proteotoxic stress, which triggers reproductive isolation. So this was the idea. So when we were doing this, I also felt that, okay, if I know that these cells are suffering from proteotoxic stress, what if I evolve it? Is there a way I can find out? I'm finding some 14 complexes. Maybe they are not probably the best solution for alleviating a cell or making a stable hybrid. There must be some other way, or maybe these are the ones. I don't know what it is. So I used 32 degrees because that was one of the conditions, and it's an easy condition in which I can do an evolution. And I started evolving these two replacement lines, 16 and 8 plus 15, which were extremely defective, and wanted to see how it goes. So when I was doing this, this is a 2,400 generation data. I evolved it for 7,000 generations, but I chose an earlier time point because it was fit enough and it was easier for me to sequence than taking a later time point which will have more mutations, and it's more difficult to decipher what is happening. Okay, so this is the evolved line, 32 degrees. So shorter the bar, I forgot to tell this, shorter the bar would mean that better fitness, longer the bar is more fitness because it's doubling time. So longer doubling time would mean that it's going slower, so it's sicker. Okay. So this is the ancestral 32 degrees. And if you look at the colors, the black is haploid 16, and the gray is diploid 16. And you have, this is 8 plus 15 haploid, 8 plus 15 diploid, cerevisiae haploid, cerevisiae diploid. Okay. So if you look at this, you can actually see that the diploid cells are more sicker. Clearly visible that 16, for example, 16 diploid is more sicker, as well as 8 plus 15 diploid is more sicker. And after evolution, it is almost the same as the wild type. This is after 2,400 generation. So though I evolved it in 32 degrees, I wanted to push it ahead and see whether it actually has adapted to other conditions or not. So I have used multiple conditions. I'm showing 3 major conditions, 2 other conditions which I have tested carefully. One is 34 degrees, which is higher temperature. So 32 degrees to 34 degrees. That's a kind of no-brainer experiment which I did. The other condition which I wanted to use was condition medium, in the sense that you culture the cell, yeast, and then filter it so that there are no cells in the medium. Now this yeast would have used up all the nutrients in your medium, and would have also secreted some metabolites. So use that medium in order to culture it and say it's like a kind of nutrition depletion medium in which you can test. So though I culture it with 32 degrees, I find that even in nutrition depletion, you still have a better fitness. Okay, so it's like, this is not the first time people are finding that evolution experiment in one condition is giving a beneficial effect in other conditions. It's not like I'm the first guy to do that, but I found it interesting that, okay, I evolved in 32 degrees, which is a proteotoxic stress condition according to me. And it also gives an advantage that later on, even if you put it in lower nutrient condition, it still is adaptive in the sense that the fitness is improved there also. Okay, but then the way they have adapted is very different. So there are two different replacement lines, right? One is 8 and 15 together, the other is 16 alone. And 16 was very, very sick. 8 plus 15 was sick, but not as sick as the replacement line 16. So this basically is the evolved sample. And these are the fitness. Basically, all of them are improved. So I've just put pluses. Now, rho 0 basically means is its mitochondrial function active or not. It's called petite cell. Rho 0 basically means a petite cell. Now, if I say this, I had used 8 independent cultures to do my evolution. I find that in the diploid case, there are 3 of them which become petite. So there is some mitochondrial-nuclear incompatibility or something which has arose during this time, and you find that they can't grow on glycerol plate. Okay. The surprising one was the 8 plus 15. All 8 of them I evolved, all of them are petite. So even in haploid, 6 out of 8 are petite. So it basically means there must have been a weak incompatibility sometime before, and it just grew stronger, which was weak initially, which was not identifiable, because when we are doing these experiments, we try to take the best colony every time from the frozen stock and keep doing our evolution. We are doing our experiment. We don't take old colonies. Now, the moment we let it grow old in rich medium, now all those weak incompatibilities somehow become stronger here, and that could be one reason why this whole mitochondrial failure or petite formation has happened. Okay, so since this is more complicated, and because it is having 2 different kinds of phenotype right now, one it being petite, and then there's a fitness improvement, though it's petite, I didn't want to deal with it at this point of time and chose the 16 replacement line. So I sequenced it at quite a high depth, 100x depth, and analyzed the data. I'm not going to the details right now because we don't have the time for that. But one or two things which I found out was that among these haploids, among the 8, there are like 2 or 3 of them which have become diploid. The haploids have converted to diploid state during evolution. And this has also increased the SNPs. When I look at the SNPs, there are like 200, 300 SNPs there. So that's one case. And people have shown, Sarah Otto, for example, has shown that if you take a haploid cell and you evolve it, it converges to diploid state. Diploid state converges to diploid state. So this could be one reason. But in hybrid, same thing could happen. So how it has converted, I'm not sure. We also don't know how it converges to diploid state every time, but somehow they do that. And the other interesting fact which I found was that though, probably I told that, though they have become petite, they have improved their fitness. So I'm really curious. Usually when you have loss in mitochondrial function, the cells get sicker. Even yeast get sicker. They are sicker than the normal wild type cells. But this has somehow increased its fitness, and I'm curious to find out what were the changes which led it to increase its fitness despite losing its mitochondrial function. Okay. So when I started analyzing this data, I also realized there were a couple of things which I had not seen at the point when I started doing my evolution. So when I did my evolution experiment, my main aim was to find a way in which it can alleviate proteotoxic stress, so I used 32 degrees. Now, heat, for example, higher temperature is a very general stress. It's also known to be a non-specific stress. It can change many, many things. So instead of that, so I now started an evolution experiment altogether in which I don't want to use 32 degrees because there is already an intrinsic stress there due to the presence of foreign chromosome. So I wanted to only evolve in the presence of foreign chromosomes continuously without any other stress, the stress only being the intrinsic stress of the presence of foreign chromosome, and see how it adapts. So I want to do it in 2 ways, one with the glucose and the other with glycerol, because now I'm seeing that the cytonuclear incompatibilities are propping up more and again and again. If I use glycerol, I don't give it a chance for that to happen. So if I don't allow mitochondrial-nuclear incompatibilities to pop up, they must probably use a different trajectory, or is it going to use the same trajectory? So this is the question which I'm asking right now. So although I'm analyzing the previous data and trying to understand what is happening, I felt that if I probably get a solution to this, this can also tell me like how does the mitochondrial, if you are not allowing its most preferred way, what it be? I mean, some kind of something like that. I want to see by looking at these two conditions. So with this, I find that the idea I started doing this evolution experiment was like the Linsky's lab experiment. Michael Desai started doing a long-term evolution with yeast. I wanted to do it with hybrids. So initially I started off with all the replacement lines, but I found that it was very difficult for me to handle because I was doing other experiments also. And finally, I chose these two because they were the sickest one. And let us see how it goes. Yeah. So this is basically what did. I would like to thank Junyi Liu, who was my postdoctoral advisor in Academia Sinica. So I was at Academia Sinica, Taiwan. And Shinyi Li, we call him Aka, he is the one who did all the biochemistry experiments. I did all the mol bio and genetics experiment, but all the biochemistry work, the protein complexome work was done by him. I just wrote the programs to analyze the data. So, and money was from my postdoctoral grant and Sinica. So thank you very much. And there's just one announcement. I've just started my lab. So anybody who is interested in working with me, I work on these areas: speciation, hybrid vigor, evolutionary dynamics of stress response, and also do a lot of computational work on population genomics. If you guys are interested, want to do a project, or you want to be a PhD student or a master's student, you can always come and talk to me. Thank you very much.